Okay, if we look at the general transformation protocol for Agrobacterium, you will have some sterile explants that have got dividing cells in them. If you don't have sterile explants with dividing cells in them, you're wasting your time. And it's usual to start off with an overnight culture of Agrobacterium. As I said here, Agrobacterium rhizogenes. Tumefaccians, disarmed strains of tumefaccians tend to be more widely used. And the transformation protocol consists of a number of steps. And the first one is a co-inoculation. So you have your dividing cells and you have Agrobacterium, which has been grown up. Now, it is usual to treat your overnight culture of Agrobacterium in order to effect the inoculation. Your overnight culture of Agrobacterium is going to be problematical in a number of ways. Firstly, the Agrobacterium will stop growing because it's run out of nutrients, and you want it to be in a good physiological state. So it's usual to get rid of the overnight medium and put the Agrobacterium into some medium in which it can grow. The second thing is that the Agrobacterium will also contain, or the medium will contain, a number of components that are shed from the Agrobacterium. So things like lipopolysaccharides from the cell wall of the Agrobacterium will be in the culture filtrate. These are not nice things for plant cells, so you want to try and get rid of them if you can. So before we use the Agrobacterium culture, it's usual to actually spin down the cells, pour off the culture filtrate, resuspend, wash it, resuspend again, and it's usual to resuspend the cells in a plant cell culture medium. So it's going to be plant cell culture friendly. The inoculation density can also be critical for transformation. So we normally do some estimate of cell numbers, cell density in Agrobacterium, and adjust the cell density to a suitable cell density. And then you will co-inoculate. So you're putting Agrobacterium and plant cells together so that the bacteria, the Agrobacterium, can attach to the plant cells. And this typically takes minutes to hours. So I've never seen a co-inoculation protocol which is more than a few hours in length. And it can successfully happen in just a few minutes. Once the bacteria have attached to the plant cells, you want to get rid of the unbound Agrobacterium. If you don't get rid of them, they will overgrow the culture, because you're going to have to do a co-cultivation for a number of days. So it's usual between the inoculation and the co-cultivation to include a wash step to get rid of unbound Agrobacterium. So after the inoculation step and the washing, you want to co-cultivate for a number of days. In this co-cultivation, the virulence genes will be activated and the tDNA will be transferred to the plant cells. So you have transfer of the tDNA. So that's everything that's in this red box at the top. Okay, so you've got transformed plant cells. You then need to get whole plants back from the transformed cells. So the first thing you do over on the right in the blue box at the bottom is you transfer to a medium that's got bactericidal antibiotics for a number of days to kill off the Agrobacterium. You're then going to transfer to a medium that's got bactericidal antibiotics plus your selective antibiotics. So if you were selecting on neomycin phosphotransferase 2, so you've put in the MPT2 gene, which is going to promote phosphorylation of kanamycin, this is the stage at which you add the kanamycin. And it's usual to actually have a selection for months. So you're killing off any residual Agrobacterium and you're selecting the plant cells which are expressing your plasmid. And you're assuming that if the neomycin phosphotransferase 2 gene is being expressed, then your gene of interest is always being expressed. It's not always the case. It depends on how successfully the entire plasmid is actually transferred and expressed. Then you're going to transfer to regeneration medium so you can get whole plants back. This is absolutely critically important. Unless you can actually get whole plants back from single transformed cells, you're wasting your time. Okay, so for protocols involving naked DNA, biolistics are now routinely used. DNA cut particles, typically a micrometer in diameter, something like that, are coated with DNA and they are literally blasted into cells by an explosive charge. I've mentioned silicon carbide fibres. This does work. I've done it myself in my own lab. There are two major disadvantages with using silicon carbide fibres. One of them is that silicon carbide fibres resemble asbestos, so you can end up with things like mesothelioma, which is a very aggressive cancer, lung cancer, if you're not careful in how you handle the fibres. And the second problem is that transformation with silicon carbide fibres uses quite large amounts of DNA. So you'll be talking about using milligram amounts of plasmid DNA per transformation. It's quite a lot of DNA to actually prepare, so it's expensive to actually prepare the DNA. So biolistics is now very heavily used in order for transformation using naked DNA. Protoplasts are cells without a cell wall. So all you do is you take your cells and you enzymically degrade the cell wall using cell wall degrading enzymes. And then you get DNA uptake either by electroporation or by treatments which change the surface charge on the plasmalemma. Things like polyethylene glycol will cause DNA to be taken onto the cell wall, and then these can be phagocytized inside the cell. The big problem with using protoplasts is it's very difficult actually to get regeneration. Even cell division of protoplasts can be quite difficult. So I tend not to work with protoplasts. They're inherently difficult to work with. And this is what a particle gun looks like. So you've got DNA coated on pellets. It's forced down the barrel of a particle gun by gas pressure. You typically need a gas pressure of greater than 1100 psi, which can be achieved by either a burst membrane or by an explosive discharge. Most machines nowadays actually rely on helium gas pressure. And the gas pressure is suddenly released because you include what's called a burst membrane, which bursts depending on the gas pressure. And the particles are forced through the cell wall and the DNA is released into the cytoplasm or the nucleus. In order to get whole plants back from single transformed cells, there are only two methods: somatic embryogenesis or adventitious shoot formation. I've already mentioned adventitious shoot formation when I was talking about micropropagation. So for example, for micropropagation of coffee, you first kick the cells into an uncontrolled cell division to produce a callus, and then you regenerate whole plants back from these dividing cells through adventitious shoot formation. So there were no preformed meristems there before you induced the callus. And this is typically stimulated by equimolar amounts and quite large amounts of cytokinin and auxin. The other method of doing it is through somatic embryogenesis, where multiple embryos are formed. And there are three types: proembryonic masses, cleavage polyembryony, and secondary embryo formation. And it's very important when you're looking at protocols for plant cell transformation that you're able to identify: are they regenerating via adventitious shoots? Are they regenerating via somatic embryogenesis? Somatic embryogenesis from proembryonic masses is the only way you can get embryos formed from somatic tissues, and it only works in umbelliferæ. This is parsley and carrot. So what happens is you can take leaf strips and you add high levels of auxin. The high levels of auxin lead to high levels of this polyamine putrescine, and this kicks these cells into proembryonic mass formation. And from these proembryonic masses, single cells are sloughed off the surface. So while you maintain high levels of auxin and high levels of putrescine in the culture, you will get cycling of cells as proembryonic masses. When you remove the auxin, you get proembryonic mass conversion from single cells to globular embryos. And this is associated with interconversions, putrescine being converted to another polyamine, spermidine. You can actually inhibit this transformation of single cells into globular embryos by inhibitors of this conversion of putrescine to spermidine. So polyamines are intimately involved in this conversion. Also, globular embryos can be converted to heart-shaped embryos who will naturally develop, but only if you have this conversion of spermidine to spermine. And again, they're inhibitors of this interconversion of spermidine to spermine. If you include these inhibitors in the medium, you can actually arrest embryo development at the globular stage. They will not form heart-shaped and subsequent torpedo-shaped embryos. But it only happens in carrot from somatic cells. It can happen in embryogenic tissues of monocots and some conifers, but it doesn't happen in somatic tissues of monocots and conifers. So for the vast majority of regeneration through embryos, in all species, apart from those in umbelliferae, that's carrot and parsley family, if you want to go through embryos, you have to start off with embryos. In conifers, they use something called cleavage polyembryony. So for example, if you look at a developing Sitka spruce embryo, as developing Sitka spruce seed, you will find that it starts to cleave lengthways. So you get lateral divisions, and you end up with multiple embryos in your developing seed. Now in the seed, the development of each embryo is limited by space and it's limited by nutrients. So one embryo will actually survive at the expense of any others that have actually been produced. So in Sitka spruce seeds, you only get one embryo that survives. However, if you take that developing embryo and put it into a liquid medium, where there's abundant space, where there's abundant medium and nutrients, you can get an uncontrolled proliferation of embryos. You can end up with millions of embryos in a 250 ml flask. And this can be a very efficient method for micropropagation. Now you are starting from seed, so you do have to have sexual recombination of genes before you can actually get an embryo formed. But it can be a very efficient method for micropropagation. And these embryo cells can then be transformed. Another way of getting embryos is through secondary embryo formation. So cleavage polyembryony only happens in conifers. Secondary embryo formation occurs in most dicots. So you start off with an embryo, and that embryo can either form embryos on its surface, or you can get the formation of an embryogenic callus. And this typically occurs with high cytokinin concentrations. And once you've actually got embryos being formed on the surface, you can then get these to form torpedo-shaped embryos. So in summary, there are many ways that plant biotechnology can be beneficial. A wide range of traits can be put into plants. Benefits of GM crops are such that the technology has been taken up very quickly. So as I've demonstrated with GM soybean, GM corn, GM oilseed rape, there's canola in the States. But we have to benefit the potential benefits with potential risks and assess release on a case-by-case basis. And some people would say there's such a thing as the precautionary principle. If there's any danger associated with release of these GM crops, you shouldn't do it. And I'd say if you take this approach, you wouldn't actually get out of bed.